I'm going to talk today about a subject I know I was going to have to get to eventually, so I've decided to go ahead and go for it. And that is the implementation of automation in microbiology, which microbiology has been totally anathema to automation for years. I do have to do a bit of a disclosure here. We are a WASPLab lab. I'm going to talk about that quite a bit. And we do have this little relationship with Beckman Coulter. So I don't want to sound like I'm trying to get you all to buy a WASPLab for your home office. But do keep that in mind. Automation in the laboratory is something that's not unusual. It came a long time ago, especially to something like chemistry. This is a picture here of our chemistry lab here at CPA. And it's hard to tell from this picture exactly what's going on, but this is a big honking machine here, and the techs will receive specimens over here on the backside. They'll put them on this machine. You can see a few tubes sitting right in here. It'll read the barcode. It'll know what tests have been ordered. It puts it on this cute little conveyor belt, which just runs around here like a racetrack. And then routes them on back here to machines that are this way. It'll put it right on the machine that's going to be used to run that test. So, the people really are just kind of putting tubes on and then verifying the results when they come off the other end. They're not actually doing the chemistry here. Hematology, pretty much the same way. This is not CPA lab. This lab is way too clean. This is not a real laboratory, I'm sure. But this is hematology. So you can see all these tubes here, these blood tubes, they go on this conveyor belt. It shunts them on down to various machines down here that will do your CBCs on those. All very automated. In contrast, this is microbiology. We have been stuck in the 19th century for a long time. We basically do all the same procedures that Louis Pasteur did. So there hasn't been a whole lot of progress in microbiology. Okay, so it's not quite as bad as that picture looks. We have done some automation. We've automated incubation of blood cultures. We put the bottles on these machines and it detects growth and they come off and we can use them for that. If we were doing this the old-fashioned way, which is where you'd look at the bottles and 2 or 3 times you would have to blind subculture. You'd have to streak every single bottle off into place, whether it looked like there was growth or not. I'd have to add 5 to 10 full-time staff just to handle doing those blood cultures. So this is much, much easier to do. We've sort of automated susceptibility testing using instruments like this Vitek or this Microscan. They go on there, it reads the results. It's a small cartridge kind of test. It'll read the results, it'll report the results out. If we were doing this the old-fashioned way, I'd probably have to have 10 or 20 people who did nothing but susceptibility testing all day and twice as much lab space in order to do it. So this has been a godsend to be able to have instruments like this. And we've gone with molecular. This is largely automated now. This instrument up here in the upper left corner is the instrument that we got for COVID. At that time, we were doing 1,000 PCRs a day for COVID. 10 years ago, just 10 years ago, if you asked us to do 1,000 PCRs a day, we would have had to hire at least 10 more people, 10, 15 more people just to do those PCRs. We were able to actually cut the number of people doing PCR. We used to have 2 people that did PCR all day. Now we have 1 person does PCR a day, and we do probably about 20 times more PCRs than we used to. So the automation has been fantastic. But what about culture? You see, culture really hasn't changed much. Excuse me. This is Karen, and she's standing there. She's doing a gram stain basically the same way that we have always done gram stains. Pouring your little bottle of crystal violet on there, and that's it. This is Lily. She's streaking out plates like we always have. We can sit there, we streak out plates for hours. Once the plates have been streaked, they go in the incubator. Here's Morgan putting a batch of plates into an incubator. And then once a day, we would take those plates out, and somebody like Nicole here would look at those plates and figure out what we needed to do to work up those cultures. Then when she was done looking at those cultures, we put them in a rack and we put them back in the incubator, and we wouldn't look at them again until the next day. And that's the way microbiology was always done. But we've kind of moved into some automation. Let me tell you a little bit about introduction of automation into the microbiology laboratory. It kind of started with gram staining. And the reason for that probably is because our pathology friends who do a lot of staining and our hematology friends who stain all those differentials, they were used to this. They were used to just taking these slides and having some kind of automated stainer to work on it. And so we came up with instruments like this. This is actually one of the newer ones that is used to automatically stain gram stains. It's kind of like a little centrifuge inside. You slip your slides in there and tell it to go, and it will stain for you. So that's a possibility. We actually used to have one of these. We got rid of it. Our techs didn't like it. You lose some of the ability to adjust staining for the how thick the smear is or what type of smear it is. So, but a lot of laboratories will now have these automatic gram stainers. What about plate streaking? Plate streaking started off with an instrument similar to this. This is what's called the isoplater. It does this circular pattern streak, and that might look really kind of cool. You might think this is a great idea, but this instrument really didn't catch on a whole lot because the only thing that was automated was the actual streaking. You actually had to apply the specimen to the plate. Then you had to mark on the plate where the specimen was so that the machine knew that. Then you had to load the plates onto the machine with the specimen already on there. It would streak it, and then you had to offload the plates. So sounds good, but it actually was not a time saver at all. As long as you're putting the specimen on the plate, you might as well just go ahead and streak it and save some time. So never really caught on a whole lot. That was then switched to something like this. This one actually was used quite a bit here. And you can kind of see it's the same pattern. It uses this cone here that the specimen goes on, and then it goes around in a circle, and you get these circular patterns, which are really kind of cool. You have to kind of modify how you work these up because, you know, it's a little different saying scant growth versus heavy growth or whatever. But it was a cool little system to do. This one would inoculate the plate for you. You put the specimens in this little drawer here, and it would take a pipette and put the specimen on right here, and then it would streak it. So you didn't have to apply the specimen to the plate. The problem with this one is that it couldn't take the caps off. So whatever tube or container you had, it could not take the cap off. You had to uncap all the specimens, put them in this drawer, and then when the specimens came out the other side, you had to recap them. But you had to be careful. You couldn't put, you know, Mr. Jones's cap on Mrs. Smith's specimen. That would contaminate it. So it caused some problems like that, which brings up a big point for all of this automation. Whenever you're going to start using these automated plate streakers, you have to have liquid specimens. That's going to be a big point. So we have moved away. We used to use a lot of these wrapped rayon swabs. You know, everything came on those. And we would sit there when we put the swab on the plate and manually streak it out ourselves. The plate streakers aren't going to do that. We have to switch to these, the liquid swabs. This is a flocked swab. It holds more specimen. It releases the specimen in liquid. So we're not actually using the swab to inoculate the plate. We're using the liquid that's in there. So an automated plate streaker would be able to utilize that liquid. Same thing with stool specimens. We don't want the actual stool. We have to put it in a transport medium, like Carey Blair medium like this, so that the instrument could actually go in and pull up some of that liquid, not trying to streak directly from a pile of you know, fecal material. And sputum. Sputum is not, you can't just stick a loop in sputum and streak it out. It's got lots of mucus and goo and stuff in it. So what we do with the sputum is we take this little device here, which is like this little corkscrew, and it screws down into the sputum. It picks up goop. They're all inside these little grooves. And then it goes into a tube that contains a mucolytic agent. It's going to digest all that mucus, and now it's liquid. So we can put that directly on an automatic plate streaker, and it will be able to streak it out. So let me tell you about the two systems that are really out there that are going for full lab automation or total lab automation. That's what they're calling it, okay? This is a Keystra instrument. It is produced by Becton Dickinson. And here's the way this thing operates. Down here, you have an inoculation, a machine's going to inoculate your plates. This one comes with a hood where you can manually inoculate plates as well. Once the plates are streaked out, they go on this conveyor belt, which brings it down the line and over here to one of these incubators. It's going to take a photograph of it at time 0. And then it's going to go in the incubator, and it will incubate for a certain amount of time, whatever is pre-programmed in. You can say, I want my urine cultures to go for 18 hours. I want my sputum cultures to go for 24 hours, whatever. When that amount of time is up, it's going to pull it out. It's going to image it. And then the image is going to be available on these reading stations. Notice the reading stations are directly on the line here. So a person will be sitting here looking at the pictures on the computer screen, and we'll decide what to do with that culture. If they need the plates to come out to them, they can tell the computer, bring me Mr. Jones' sputum culture, and the plates will come out of the incubator and come right to the bench top, where they will be able to do the workup on it. So that's a Keystra. Great instrument. Any lab would be happy to have one. This is the other one that's commonly available. This is the WASPLab. It is actually built by Copan, has a few different distributors in the United States, but it's the same kind of process. Here's the plate streaker. The plates get streaked. They go on the line. This is the conveyor belt, which is to bring it into one of these imaging units. These are going to take photographs at time 0. Then it slides into one of these incubators. It'll stay there for a certain amount of time. It'll come out and get re-imaged at certain pre-programmed times. Notice that the readers are not on the line over here. They can be sitting all over the place, wherever. But they will look at the pictures and they will decide what to do. If they want to look at a culture, it comes out into one of these canisters. So you can collect 10, 12 cultures at one time and then pull them over to your bench and actually do the workup on the cultures. I wanted to show you this thing in action. So I'm actually going to show you a little video here. This is from Beckman. And let me move this over here. I think you're going to see this here. Hopefully it's not too much of a commercial. I'm going to turn the sound. Sound is off. Good. I feel a little bit better about showing this because this is actually filmed in our laboratory. So these are our instruments right here. You can kind of ignore this going back and forth bit here. But just to show you what this thing actually does here. Here's Kathy. She's one of their trainers. She helped us set up our instrument. You take the media that you need. You're going to put it on this carousel. It can obviously hold lots of plates. You do have to make sure you put the right plate in the right slot. Then when specimens come in, they just go right on this little input device here. It's going to come in. The machine's going to pick it up. It's going to scan it. Okay, it knows that's a urine culture. So it's going to come over here and pick the size loops that we need for urine cultures. This is a little vortexer. It's going to mix it up so we know it's nice, well mixed. And here's one of the big developments that we can do now over that previous plater is it's going to take the cap off. They're not really showing it here, but it takes a cap off over here. There you go. Sticks the loop down in that specimen and pulls out the appropriate plates, and then it's going to streak the specimen. This is actually pretty impressive. They do a good job without digging up the media, which humans often do. So it streaks the plate out. Then, if it's a specimen that requires a gram stain, it will also make a slide. It's not going to stain the gram stain. You still got to do that yourself, but it will make the smear for it. Then that specimen's done. It's going to take it and put it back in the rack over here. Okay. The plate gets barcoded. It scans it to make sure it can read it, and then puts it on this conveyor belt. That takes off and goes down the line. Now, this is going to show you what's actually inside the instrument. We don't leave the instrument open like this, but it's going to bring the plate in here. Again, it scans it to make sure it knows which one it's got. It's going to set it right there. Then it will photograph it, give a time zero picture. Here it comes. That way, you know if there was something wrong with the plate to begin with. When we did it manually, we could tell if there was something wrong with the plate. But since we're not doing this manual anymore, we don't see that. Goes through that little bitty slot into the incubator, which holds a whole lot of plates. It'll flip it over and it'll slide it into one of these slots here. Okay, so it's going to incubate there. And like I said, at a certain amount of time, it will pull that plate out, it will re-image it, and then it will show up on our computer screens, and we can do the workup or whatever we need from that point. These are the carousels. The plates will come out. If you call for a plate, it'll come out and get put in one of these carousels. And then Kathy's going to come along here. She's going to pull out that carousel to take the plates. There she comes. So these can be plates that you want to work up, or these could be plates that you say, okay, I'm going to take these, I'm going to put them in our morgue. We're going to save them for a week, just in case physicians call back and want extra workup done on those. It's also pretty cool. If a plate's no growth, we're done with it. It throws it in the little trash there all by itself. So you never have to touch a whole bunch of these plates. On the computer screen, we'll look at the cultures. We'll decide what needs to be done with these and say, okay, we want to work up this isolate. We want to send that out. And then it will come out and our techs will be able to work it up. So that was our little video. Like I said, not meaning to really advertise for them, but that was probably the best way to show you exactly what this thing does. I want to show you some about the computer part of it. This is just a cute little dashboard, which tells you what you've got coming up. So it's 7:30 in the morning right here. This is telling you I've got 26 urine cultures that are going to be ready to be imaged at 7:38. I got 3 more urines, 8:25. So you can kind of look at the schedule and say, this is when these cultures are going to be ready for a person to actually look at those pictures. Which brings up a very important part about this. This completely changes the staffing needs in a micro lab. Like I said before, we would take these plates out. Maybe respiratory cultures, we read those up somewhere around 8 o'clock in the morning. We looked at them, okay, we did our workup, and then we put them back in the incubator and brought them out again 8 o'clock the next morning. So you were reading plates once a day. With this sort of system, you want to be able to read the plates whenever they're ready to be read. When you've got that image, once that image is there, that's when you want to read. So now, instead of saying we're going to read plates on first shift, to make the most use, the best benefit out of this instrumentation, you want to be able to read around the clock. If a plate is ready to be read at 1 o'clock in the morning, I want somebody there to be able to read it at 1 o'clock in the morning. We are not there yet. Our laboratory is not there yet. We're kind of stuck somewhere in the middle between the old way of doing it and the way we want to be doing it. But we're like a lot of labs, it's very, very difficult to find staff. Especially staff who don't mind working in a big laboratory by themselves in the middle of the night. When it's ready to be imaged, after it's been imaged, it'll show up on something like this. This is telling me I got 19 sputum cultures that are now ready for me to look at, 2 throat cultures, and we can click on these explore buttons and we'll see something like this. This is a sputum culture and it's going to show me the plates. And then we're going to decide what needs to be done with it. So if I look at this and think there's something in here that needs to be worked up, something's important in here, then I can say send a reader or positive workup. That's going to come out to one of those stackers, and then we can pick it up and actually work at the plates to do the actual identification. In this case, I'm going to look at that and say, this is normal respiratory flora. There's nothing here that I'm interested in. So I'm going to come right down here and say light growth normal respiratory flora. That is going to send the report immediately into the lab information system. It's going to get reported in the chart at 18 hours, light growth normal respiratory flora. Now notice you can see here, this is the 18-hour read. It's got another 36-hour read coming. So these plates are going to stay in the incubator and be imaged again after 36 hours. So this is going to be a preliminary normal respiratory flora report. If it's the same thing after 36 hours, then I click that button, it's going to be a final report. So notice, I didn't touch those plates to streak them. I didn't touch those plates to read them. Only if there was a positive workup that I actually needed to touch the plates. And there's one other cool thing about this. Here's 6 urine cultures that are ready to be read. You'll notice this little wand over here. This is telling me that there are 2 cultures that the machine has already decided are no growth. So I can click on that little wand there and it shows me this. Here's urine cultures. These are 18 hours. This is a final read because non-invasive urines get read at 18 hours. That's our criterion. And then they're done. So I just scan down the screen. I say, I agree. Those are all no growth. I click on this send button and the final negative report goes immediately into the computer without my intervention at all. And those plates actually come out and get thrown into the trash automatically. So I figure, we do somewhere around 300 urine cultures a day, and about 1/3 of those are no growth. So this is 100 cultures a day here that we don't touch the plates to streak it. The plates go directly in the trash without us ever having touched the plate. There are some other algorithms. There's one for mixed growth urine cultures where it looks at it and says, this is a mess, this is not real. So we don't have that one yet. And various chromometers. MRSA surveillance algorithm, we have one for that, which is great because we do about 100 MRSA cultures on the NICU every Monday, and 99% of those are no growth. So this machine tells us these are the ones that are no growth. Click this one button, and they all get finalized in the computer system. They all get thrown away. You don't have to touch them. You can imagine how much time that saves. Excuse me. So let's look at that. Are these things really any good? This is a great paper that came out a few years ago. These are 4 different hospital laboratories or system laboratories that switched to the WASPLab like we have. And they got together and they started looking at how much productivity did we actually get out of this? How much increase in efficiency did we actually get? So let's look at a couple of things here. This is before they had WASPLab. You can look at how many specimens per FTE total. And you can see that's how many specimens one person could work up in one day. Seems kind of low to me, but okay, I'll go with that. After WASPLab, everyone had this nice increase in the number of specimens that they could work up per FTE. Okay, if you looked at the labor cost per specimen, there was a decrease in labor costs per specimen in every one of these laboratories. May not seem like huge numbers, but I kind of figured it out. If our laboratory saw the same increases in labor costs per specimen, it would save our laboratory about $400,000 a year. So yeah, that's a significant savings just in labor costs there. And notice that every single one of these places also had a significant increase in their volume. This is how many specimens they saw per day. So they all had this nice increase in volume, which is something that's really very important with these. I'll tell you why later. This is the same paper. They looked at turnaround time. This is just urine cultures. I don't like the graph at all. I don't like the way they did that, but the important thing to look at is the change from this blue line down, which was before, to the purple line, which is after full implementation of the whole thing. So there was a decrease in turnaround time for urine cultures. And I think the important thing is looking at this right here. They went from 16% that could be finaled in 24 hours, in less than 24 hours, to 58%. So 16 to 58% finaled in less than 24 hours. Now, honestly, the only ones that are going to be finaled in less than 24 hours are those that are no growth or those that are mixed flora. If there's something that's got to be identified and worked up, it's going to take more than 24 hours to do that, which is why you get this second hump over here. So even with those that require identification, you're getting faster turnaround time than you did before. So here's some more papers that were done on turnaround time with one of these two systems. This showing you a significant decrease in turnaround time for blood cultures and body fluid cultures. And this one going back to urine cultures, this is a median turnaround time, which again, is largely going to be determined by how many no growths or mixed floors that you have. So what we see with these, the data is showing that with these instrumentations, you do get better efficiency of working up cultures. You save a lot of time, you save a lot of tech time, and you get the results out faster. So to finish that up, let me go with this paper. This is a paper that was written by Tom Thompson, who runs the laboratory at North Shore in Chicago. They were the first lab in the country to have either one of these instruments. They got a Keystra when it was fresh off the freshly available. So Dr. Thompson wrote this paper summarizing the whole thing. So here are the gains. This is what you get out of these instruments. Automated barcoding. It's going to label the plates. You're not going to have all these mislabeled plates. You're not going to have incorrect media. You're not going to have misplaced plates. It keeps track of it. It's a better system for keeping track of your media. Automated plate inoculation and streaking. That machine can actually overall streak better than humans can. And I say that because it doesn't get tired. You know, when we used to have to streak out 200 urine cultures every evening at 9 o'clock, you know, we'd have two people who would sit there streaking urine cultures for hours. Can you imagine? I mean, that's mindless work. You're going to get bored. You're going to fall asleep. And you could tell people would get sloppy in their streaking. This thing doesn't get sloppy in its streaking. It never gets tired. So you have better quality streaking, and there are several papers out there that show you're going to have a better isolation of organisms using these instruments. Better use of skill label. Why are we taking somebody who's got a college degree and making them sit there and streak urines for 2 hours? Give them something that takes a little bit more brain power to do. Decreased incubation time. One thing that we really didn't mention, if you saw that video, it stuck that plate into the incubator through that little crack and that little slot that closes up. So these incubators stay closed all the time. Think about the old time when we would open up that door every 10 minutes to put plates in or take plates out. It never stayed up to temperature. It never stayed up to the correct carbon dioxide concentration. Now we have basically almost an airtight incubator that is going to hold temperature and hold carbon dioxide concentration constant. So the bugs actually grow amazingly much faster than they used to. We finalize urines, non-invasive urines, at 18 hours. We couldn't do that when they were being put in a regular incubator. Labor savings we've talked about. Reduced FTEs are really what's more important these days as more and more hospital laboratories conglomerate, join into systems, make a single large microbiology lab to cover several hospitals. What's more important here is the ability to take on more work and have the same number of people. You know, we've gone from 5 hospitals to technically 8 hospitals, almost 9 hospitals. You know, we've increased our work and have been able to do it with 4 fewer people than what we had prior to implementing automation. Efficient workflow, accessibility of culture images. I think that's a biggie. You know, you've seen, if you've come to these grand rounds here, you've seen I've shown pictures, you know, come out of that machine. I think it's just really cool to be able to have a picture of all these cultures and to keep it forever. It's great for case studies and things like that. And safety, we're not exposing our people to those bugs as much. If there's a very dangerous bug, it's going to be kept inside that machine for a little bit longer. Eventually it's going to come out, but hopefully by that time we may have an idea of what we're dealing with. This is what Dr. Thompson says for losses. Loss of technical knowledge of skills. Of course, every time we bring automation in anywhere, it means that people are not going to be doing that job anymore. I don't want to say that, not doing the job. They're not doing that little aspect of the job. So yeah, you're going to have people who aren't gram staining as much. They aren't streaking plates as much. You're going to lose some of that technical skills. This is interesting. Loss of pride and ownership with the conventional workflow. In other words, we can kind of say here that, look, chemistry was automated and hematology is automated, and we're still so old-fashioned. But we had a little bit of pride in the fact that we were still doing things the old-fashioned way. We still do it. You chemistry people, you put the button, you put the machine in, and the machine does everything. We still did all this work here. So we had a little bit of pride in that. Like I said, you want to read around the clock now. In the old way of doing things, the plate readers were first shift. And if you're going to become a clinical microbiologist, you kind of felt good about it because I'm going to be working first shift. I'm not going to have to work in the middle of the night like those chemistry people do. Well, now we're trying as best we can to find talented microbiologists who can work and are willing to work in the middle of the night. And this is the big thing. These are big, complicated instruments with lots of moving parts. They do have downtime. They do break every now and then. We're very fortunate in that we have two completely separate lines, so we don't lose more than one. We don't lose more than half of our capacity at one time. But if we only had one, I would really be kind of ticked off whenever it went down. It does happen. Now, the big question here: who can afford this? They are very expensive, very expensive. A small micro lab is not going to be able to have the space for this. They're not going to be able to afford it. Ongoing expenses high. Service is very pricey, and service is going to be extremely important. And there's a lot of IT expenses as well. Even buying some of the software upgrades is outrageously expensive at some point. So these cost a lot of money. So what are you going to save? We've already talked about hard dollar savings as far as of labor. Where you're going to save quantifiable amounts of money is going to be less labor or the ability to take on additional work without having to add FTEs. But that's not going to cover the whole expense. We don't pay our techs that much. Okay, so it's not going to cover the full cost of the instrument. So you kind of have to make the argument also on soft dollar savings, unquantifiable. You're going to have reduced turnaround time. You're going to have better quality. How much does that lead to dollar savings in other areas of the hospital? Or how much does it just result in better patient care, which is also important, not just the money. I threw this in here as well because Dr. Thompson brought it up. Lab space is very expensive. And so you really need less lab space here. You can actually have people working remotely and doing some of this work now because it's done on computer. So Dr. Thompson's recommendation, especially when you're talking about laboratories that are combining, have large laboratories that cover several hospitals, with all that's going on here, it's not a question of can you afford it or not, but can you afford not to do it? I don't expect to see these in small community hospitals, but there's an awful lot of small community hospitals that are not doing their own microbiology anymore. It's been farmed out to a larger facility. So after all these years, saying no, automation is never going to come to micro, pigs are actually flying when this comes. So that is all I have for you guys. I do see a question from Avery.
Yeah, I think there's a couple of questions. There's one from Avery there that says, how is cross-contamination prevented for all the specimens that go through the WASPLab assembly line?
Yeah, cross-contamination. Okay, a couple of places you can think about it. First of all, there's a little incinerator in there. So it's going to sterilize the loops in between plates, just as if you were doing it manually. So you're not really worried about that bit. You can look at, aerosol formation. Is there a possibility of things, being left behind? You do have some, I'll tell you one thing that does happen every now and then. It drops tubes every now and then. Usually it's not going to drop a tube while it has the cap off, because that tube does not move when the cap's off. But there's that possibility of dropping the cap, which could be contaminated. There could be aerosols inside there. What we can do is we regularly clean the inside of it, so we don't have that issue. And what we do is we'll occasionally run plates through. We'll put on a tube of sterile water and run it through the machine to show that we're not getting contamination from that way. So is it possible? Yes, it's certainly possible that there can be some contamination, but we do the best we can to control for that. Also, when you look at the plates, you know, you'll get a good idea from looking at the pattern of streaking whether this was contamination or not. Because you'll have colonies in places where you really shouldn't be having colonies, if that makes sense.
Sure. So I think that answered both the questions, because the other question was around the loops used for streaking being disposable, or do they get sterilized? And you said they are sterilized.
Yeah, for the WASPLab, it has what looks like a regular loop. Each loop can be used something like 10,000 times. So it does sterilize those in between. The Keystra does it different. The Keystra has a little metal ball that it puts on the plate, and then it runs that ball over the plate using a magnet. So it doesn't reuse anything. That ball is still sitting there on the plate when you look at it the next day. So you have to buy a lot of little metal balls to do a Keystra.
All right.
Hey, Alan, I can ask a quick question.
Yep.
Who's working on the automation between a positive plate and then getting it onto like automated susceptibility testing?
I decided not to bring that up.
Okay, never mind.
There is an instrument that's available for the WASPLab called a Calibri, which will actually take your plate and spot a multi-target. It'll pick a colony and spot a multi-target for you. And it will set up, it won't set up completely automated susceptibility testing. But for example, if we're going to use a microscan, it'll produce a 0.5 McFarland. That you wouldn't have to do that part. It could go and you can take that tube and then put it on a microscan. And I know for the other instruments, they are certainly working on ways to be able to get it to automatically inoculate those panels. We don't have a Calibri. We have no plan of getting one because it is another big instrument and we have no place to put it. It's got to sit at the end of that line there. And honestly, just don't think it's going to save us much in the way of time. So we don't see that happening for us. But yeah, there is an instrument called the Calibri that is kind of taking the next steps in doing the whole process.
Okay, thanks.
All right. Always very interesting, the workings of the microbiology department. Anybody else have any other questions for Dr. Junkins? All right. Well, I'd like to thank you, Dr. Junkins, for doing all the grand rounds that you have done for the year. Very much appreciated and always very informative. So thank you very much for your dedication to the NIDI grand rounds process. So I hope everybody has a great day, and we will see you all in November.